so what is happening inside of an infected host cell? Well, one of the really important things that happens inside of an infected host cell is that the capsid or the protein coat has to be put together. And what you can see on this slide is really I hate to say it's beautiful because it's viruses that kill people, but they are beautiful. It's this really nice geometric assembly, and there's these repeating units, and there's this incredible symmetry and organization to the way that capsids are put together. So you can see different viruses put their capsids together in different ways, and this is one mechanism that scientists can use sometimes in order to classify viruses. Because viruses are so small, all microscopy to actually visualize a virus is going to have to be electron-based microscopy. And it's generally going to be transmission electron microscopy. So those of you who might be working in a clinical lab setting will not use microscopy to visualize viral particles. You may use microscopy to visualize viral damage to host tissue. When we have a virus that is enveloped, I told you that there are some glycoproteins that are going to be embedded in the surface, and those can serve as what we call viral attachment proteins. Remember, when we were talking about bacteria, I told you a really important virulence factor for bacteria is some sort of adhesion protein. Because if a bacterium can't adhere to host tissue, it can't cause disease. It'll be wiped right out of the host. The same principle applies to viruses. If a virus cannot bind to a host cell, it cannot be internalized and it cannot cause infection. So having a naked virus, all those proteins are exposed, and so they can use those to bind to the host cell and get in. But with an enveloped virus, if the outside of the virus is basically a eukaryotic cell membrane, how are they going to get into a new cell? Well, they embed viral proteins in that membrane. And those proteins then can bind to receptors on the host cell and allow the viral particle to be internalized. So in a classic viral infection, step one is going to be recognition. The virus recognizes or is recognized by the host cell. The virus will then attach. Sometimes we say that the first step is attachment. Whatever, either one is fine. But often the proteins that are used by the virus to recognize the host cell and to attach are also the ones that will be recognized by antibodies. Once the virus has attached, we have what we call penetration. The virus gets into the host cell. In animal viruses, this is usually through endocytosis. So the virus is brought in. You then have uncoating. So remember, we're bringing in the whole viral particle. So we have to get the nucleic acid out of the protein coat. And then it depends on what type of virus we're talking about, but these steps all might be slightly different. At the end of the day, though, we're getting the nucleic acid out. We are going to transcribe so that we can translate and replicate viral nucleic acids. During translation, we're also going to be making all of the proteins necessary for viral assembly. So we'll have assembly and then some sort of lysis and release with our naked viruses. Perhaps we have making of an envelope for our enveloped viruses. And what they're trying to show you here in pink are different steps where we have drugs that can interfere with some of these processes. But again, if you find this figure overwhelming for the moment, that's totally okay. We will see figures like this that are specific for different viruses we talk about, so we can break them down for the individual viruses. Because again, when we talk about DNA versus RNA, single-stranded, double-stranded, plus sense, minus sense, some of these steps get blurred and changed. inside of a host cell, if we're talking about a viral, growth curve, we don't see that classic growth curve that we see with bacteria, where we have lag, log, stationary, death. Instead, what we have is basically the virus getting into the host cell. We can no longer detect virus outside of the host cell because it's inside and replicating. And then all of a sudden you have all of the virus come out, especially when we're talking about naked viruses. And this is called the burst size. It's basically the number of infectious particles that are released from an infected host cell. However, the majority of viral particles that are released are actually defective because they're non-infectious. There could have been errors in nucleic acid synthesis. There could have been errors in protein synthesis. And so even when we talk about having a really high burst size, meaning we have a ton of infectious particles being released, you have to think that there's going to be a lot more non-infectious particles that were also released. The time it takes an individual virus to reach its maximum burst size will differ depending on different viruses. So you see some of the viruses down here in the bottom, like the P coronaviruses, that includes things like polio, rhabdoviruses, rabies, toga virus. We're not going to talk a ton about the toga viruses, but we talk about them more in the arthropod-borne section. Within a few hours, they reach their burst size. And their burst size is huge, between 10 and 100,000 viral particles being released from an infected cell. Flu and measles, a little bit slower, fewer viral particles, but remember, every infectious viral particle that's released from a host cell can go infect another cell. And then our retroviruses are pretty slow, and they release far fewer. And you see it's a little bit like more of a gentle slope instead of this explosion. A lot of these are naked. These are enveloped. So you get that slower release. So that first step is going to be recognition or attachment. Viral attachment proteins, whether or not they're part of a naked virus in the capsid or part of an enveloped virus in the envelope, will bind to receptors on the host cell. This is the limiting factor about where a virus can go. Those viral attachment proteins are only going to be able to bind to certain receptors on certain types of cells in certain types of organisms. And so if they don't have a very broad type of viral attachment protein, they may be very limited. So we have human-only viruses. We have viruses that only infect nervous tissue, viruses that only infect gastrointestinal tissue. And so if we have only certain organisms that we can infect, we call that the host range. For example, smallpox only infects humans. That is a very limited host range. Some viruses, like rabies, can infect lots of different mammals, so there's a more broad host range. And then we have tissue tropism. Does the virus encounter cells that actually are susceptible? HIV can only bind to cells of the immune system, so it has to bind to cells of the immune system. Rabies binds to cells of the nervous system. But we have other viruses that could bind to the endothelial tissue, to some epithelial tissue, to different kinds of blood cells, so they may have a more broad tissue tropism. So these are terms that are important in virology, the host range and the tissue tropism. Again, this is a rather complicated image that we will talk about each different group of viruses when we get there. But you see naked DNA and positive strand RNA genomes are sufficient to cause infection if just the genetic material alone is injected into a host cell. We call these infectious nucleic acids. This never happens in nature. This is totally a laboratory artifact, but because positive strand RNA can act as an mRNA template, you can immediately begin protein synthesis. And because DNA can be transcribed into mRNA and then be translated, it is also just a one and done. You can get it in and get things going. With our negative strand RNA viruses, our double-stranded RNA viruses, our retroviruses, there are more complicated steps that go on. And I'm not going to belabor the point here on how DNA virus replication works, because we'll talk more about it later. But you have those same steps: attachment, penetration, uncoding, and then the key thing to remember with DNA viruses, most DNA viruses, there will be an exception, with most DNA viruses, the DNA has to get into the nucleus. Why? I want you to think about that for just a second. Why does the DNA have to go into the nucleus? The DNA has to go into the nucleus because that's where the machinery is to do DNA replication and to do transcription. So we get mRNA being produced, which leaves the nucleus and then allows for protein synthesis in the cytoplasm, just like normal. And then you have the assembly and processing and release. The other thing to highlight on this image that we'll see with other viruses as well is that we often have early and late protein synthesis. So early proteins that are being synthesized are necessary for viral genomic replication, viral transcription. Late proteins that are being synthesized are usually the ones that make up the capsid, so they're the structural proteins. Okay, DNA viruses. So what is the benefit to being a DNA virus? Well, DNA is far more stable than RNA, so you have a pretty stable genetic material. Lots of DNA viruses can establish what we call persistent infections, often because the DNA can be incorporated into our chromosomes. So we can have latent infections. This is what the herpes viruses classically do. They stay inside of the host cell until they're reactivated. They can also be immortalizing. They can cause cancer. DNA genomes are found in the nucleus, like we saw on the previous slide, with the exception of pox viruses. We'll talk about pox viruses in a little while. Viral DNA looks a lot like host DNA, so we use the same transcription and replication machinery. Viral genes have to interact with the host transcriptional machinery. That's why the genome goes into the nucleus, with the exception of pox viruses. Early genes encode DNA binding proteins and enzymes for transcription and translation. Late genes will encode structural proteins and maybe any proteins that are going to be packaged in the viral particle itself. DNA polymerases generally will require a primer to replicate the viral genome. Often we provide that for them. They may bring it themselves as well. Larger DNA viruses can actually encode proteins to ensure that the host cell during replication only replicates the virus, not host cell genomic DNA. So they can promote their own replication over that of the host. RNA viruses are going to be a little bit different. Again, I know this is a complicated figure. All I really want you to focus on at this point is that we totally avoid the nucleus. So RNA-based viruses will stay in the cytoplasm of the cell, again, with very few exceptions. Now, RNA is less stable, so the genome is more prone to mutation. However, we already saw in the previous video that the majority of viruses that infect animals are actually RNA-based viruses. So even though it's inherently unstable and it may be more prone to mutation, RNA viruses are very efficient at causing infection. Most RNA viruses replicate in the cytoplasm. And so here's the trick. RNA-based viruses have to encode their own RNA-dependent RNA polymerase. Eukaryotic cells have no mechanism to use RNA to make a copy of RNA. We only can use DNA to make RNA. And then we use the RNA to make proteins. We have no mechanism to make RNA from RNA. So RNA viruses have to encode their own RNA-dependent RNA polymerase to allow for replication of their genome. Depending on whether or not they're single-stranded positive, single-stranded negative, or double-stranded, they may have slightly different mechanisms of transcription and replication. And RNA viruses are more prone to mutation. DNA polymerase, especially in the eukaryotic cell, is very efficient at proofreading. These RNA-dependent RNA polymerases, less so. Again, the genome structure and polarity determine how RNA is generated and proteins are expressed. RNA viruses, except for a plus-sense RNA genome, have to carry polymerases because, again, they can't copy their own RNA. And all negative sense strand RNA viruses that have been discovered are enveloped. So I say that have been discovered. Who knows? Someday maybe they'll find a negative sense strand RNA virus that is not, but at this point they all are.